So Stilla Technologies is who we are. So we're a startup company, but we're already 11 years old. We're based in Paris. What we do, the reason why we exist, our mission is to improve healthcare by providing solutions to high precision genetic analysis. And in particular, we do that by providing tools, instrumentation, assays for digital PCR, the new generation of PCR test. And so as a company, we were born in 2013. I created the company back then out just after doing my PhD. And we went from an idea on a piece of paper to where we are today by building 3 generations of our digital PCR systems, putting them on the market, having them used by hundreds of customers, improving along the way. To do that, obviously, we needed to have funding. So we've raised about 80 million euros of capital from VCs to fund that story, and that is still going. And we're already present globally, having customers mostly in Europe, in US, but also big activity in Asia and China and Japan, for example, across many different fields. So we are in the field of PCR. You know, PCR is used for just about everything in biology. And so our customers also do diagnostics, but can do research, can do quality control in pharmaceutical. So there's a lot of different applications. Next. And so the reason why I started this company at the beginning, and the reason we still do what we do, high precision genetic testing, is because we know that there's a huge potential to look deeper, to look into more details at the biological samples that all biological companies, labs work on. Just to illustrate that potential, if you think about those 10 ml blood tubes that you get from the doctor's office, in those 10 ml of blood, you have just about as many DNA molecules as you have trees in the Amazon forest. And that analogy is true to represent the quantity of genetic material, but also the diversity and the complexity. And it just means that pretty much in a blood of tube, you have genetic information about just about any disease that a patient can have. But in most cases, this genetic information will be present in trace quantities. So you need to have sensitivity, precision, in order to understand the potential diseases of the patients. So there is a real need for this high precision genetic testing. And at Stilla, maybe next slide, we apply this concept of high precision genetic testing, not just for diagnostics, because as I was saying, PCR can be used for many different things, but also for other fields like cell and gene therapy, pharmaceuticals, applications, etc. So our main market for high precision genetic testing is indeed, diagnostics on the 2 sides of this slide. The big application that we've been pushing from the start has been liquid biopsy, the idea that you can have information about the cancer of a patient from a simple blood draw. And this is really the best case study of why it is important to do high precision genetic testing. Because obviously, you want to detect those cancer biomarkers when they're present in low quantities, when the disease is still small and treatable. Cancer is a very complex disease, so it's not just one biomarker. It's not a simple COVID test. You need to look at different mutations, so you need to do high multiplexing. And you want to quantify also. So you just don't want to detect, but you want to know if you find a lot or a lot more than the previous time to know the disease has evolved. So this is really the best example of why high precision genetic testing is important. But I know here in this room you may have, around in this campus, in the biopark, here you have companies in pharma. And in pharma, cell and gene therapy, most modern pharmaceutical compounds are actually becoming very complex from a genetic standpoint. And so there's a lot of applications where you need high precision genetic testing for pharmaceutical companies to test the purity of cell and gene therapy products, whether they are the right ones, whether you're going to change the DNA of the patient in the way you want it, etc. So you find applications around different fields. And so at Stilla, the way we do high precision genetic testing is by doing digital PCR. So I'm sure everybody knows about PCR. We all do since COVID. The whole population does. But in digital PCR, what we do is that instead of doing the genetic analysis in the bulk of the sample, where you have all the different DNA molecules that are mixed together, we start by partitioning, cutting up the sample into tiny compartments, so tiny droplets. And then we do a PCR on all those tiny droplets. And this partitioning step, it does a very physical mean of separating the different DNA in different compartments. So in each droplet, you will have one DNA molecule of different types, such that at the end of the PCR, you can just look at the color of the droplet to know what type of DNA is in each droplet. And by counting the number of blue droplets, you count the number of, cancer biomarker 1, you count the number of green droplets, you count the biomarker 2, and you can really count a single positive droplet, meaning a single DNA molecule, and you can have a very good precision because you can differentiate between, let's say, 100 and 110 positive droplets, so to quantify very precisely what is in your sample. So this is the concept of digital PCR, which we have developed to the most advanced and accessible way today with our latest product. I think is on the next slide. So in practice, we make this crystal digital PCR technology accessible to our customers by providing a full solution, meaning the instrument, so this is what you see here on the left, the NIO system. It's an instrument similar to a qPCR instrument where the user will just put his plates to be analyzed, and then we will perform the digital PCR analysis inside the instrument with just the press of a button. So from a user standpoint, it is a very simple, accessible solution. Anybody who is doing normal PCR can become a digital PCR expert with our solution, pretty much, in a day of training. But the instrument only will not be sufficient for our customers. We also need to provide the reagents, the tests. And so we have actually developed master mixes for digital PCR, but we also have developed assays and approaches in the field of PCR to expand the multiplexing capabilities way beyond what has ever been doable in the field of PCR and genomics. With an approach called color combination, we can test up to 20 different biomarkers in a single test, which is absolutely unique. So we're, in a way, bridging the gap between traditional PCR and NGS. So PCR, typically you test a handful of biomarkers. NGS, you screen hundreds. We can provide a single test to screen 20, and with a small panel of these five 20-plex assays, you can have 100 mutations, 100 targets screened in just an hour and a half. So we're bridging that very important gap. And our products are already in the hands of many customers. As I said, about 400 customers. We're already starting to make an impact on how healthcare is being implemented in some institutes. So this is a very nice case study of CHU Pontchaillou in Rennes, in France, where Dr. Alexandra L'Espagnol has adopted our digital PCR approach to change how she stratifies cancer patients and chooses which drug to administer to patients. Before, they were doing NGS all the time, but NGS is costly, and it takes several days to get a result. And so instead now, she is using our high-plex digital PCR solutions, which will give a positive results for not all patients, because we only cover 10, 20 different cancer biomarkers. But for, let's say, 70-80% of cancer patients, she will have a positive result in a single day. She will already know which treatment to give to those patients. And for the 20% that remain negative, she will use NGS to find the more rare mutation that that patient has and make the right clinical choice. But by combining NGS and digital PCR, she is improving the stratification in terms of time, in terms of cost. And I think this is very exciting for us to see that we're already making those changes. And we also have other partners like bioMérieux, infectious disease company, obviously, who really believe that digital PCR will make a strong impact in the future of diagnostics in the field of infectious disease. This is more later stage. They are starting to work with the technology and invent the future of respiratory disease diagnostics. And again, we are in those two fields, 1 foot in the research and 1 foot in the diagnostics, and trying to work on both at the moment to empower as many customers as possible. So that's what we do, the short story. And then the last part of my presentation today is to tell you a bit more about the journey, the story behind Stilla. So I created that company 11 years ago. And it's been 11 years, obviously, of many successes, but also many challenges. It's a long and bumpy road. If there are some entrepreneurs in the room, I'm sure you know that. But so we've launched 3 generations of digital PCR systems in those 11 years. We have already a technology that's serving customers, doctor, patients. We have about 400 of those companies, rate 80 million euros. And it's been a very enriching journey for me, and I think for anybody who's worked at Stilla. We have a very multidisciplinary, multicultural team within Stilla. That's a great environment in which to work with. But that's the fun part. That's the nice part. Obviously, you also have all the challenges. And if somebody wants to talk to me about those, I'm always happy also to share my experiences about the failures, about what we did wrong. And so we've had a lot of issues along the way. We sometimes had to I had to manage the company with very limited cash visibility, which is the reality of many startup companies. You never have a 5-year budget in front of you, not like in the top 10 biotech companies. That doesn't happen at Stilla, not yet, at least. We've had to manage lawsuits and IP lawsuits in the US, which we prevailed in the end. So that was a long story, but turned out well. You always have the issues of hiring the right people, and that's, I think, one of the most important and most challenging action as a manager, as a CEO of a company, to get the right team together. And obviously, I think Ooho will talk about that. It's a very different story to invent products than to sell products. And so I, as more of a researcher, I've had to learn how to do things differently. I've been successful in inventing, building products, and I've learned how to be more successful, or at least do less poorly in selling products over the years. You can go to the next slide. And so a few slides about the long road from innovation to product from a technology standpoint. So it all started during my PhD, 2009, 2012. And in the beginning, I had a successful PhD, so a bunch of published papers, 4 patents, but I was doing a PhD in microfluidics, very theoretical, not applied at all. And so after 3 years of my PhD, I started thinking, okay, what can I do with this technology? It can be used for many different things, but it would be nice that it doesn't end up just being a patent from the university that's just sleeping in a drawer, as there are so many in universities. And so I spent quite some time thinking about the applications, and then I thought, okay, there's this very nice application, this technology of digital PCR that is very hard to implement in labs today, has a lot of potential. I believe my microfluidic technology can make it more accessible, simpler to implement in the labs. That was the starting point. But I'm a physicist, more by training, engineer, so I have no idea what molecular biology is about. So that led me to the second phase in the project, which is the year of creating Stilla. When I start, I'm alone. I'm a physicist. I don't know anything about genetics, PCR, or business, so I need a team of co-founders. I've done my PhD, but that's all my professional experience, and I was not paid a huge amount through my PhD, so I have not put aside money that I can invest in my company, so I need to get cash somehow. And I've never done digital PCR, let alone PCR in my previous life, so I somehow need to get this thing to work before I create the company. And so that's what I did during a postdoc, addressing those 3 challenges, and then decided to create Stilla when I had tackled those 3 first items. I had a starting team, a starting amount of cash, and we had done a digital PCR, and we said, okay, this can work now. We just have to make it better. And so that's the next slide. We had to turn the first products, the first prototypes made by ourselves, like a DIY digital PCR system on the left. We had to turn that into a product we could sell and that any user could use, and it took us about 3 years to create this most simple viable product that we started to sell. And so maybe just a small sharing of experience and lessons in those 3 years. One thing that I think we did very well was to focus our attention on what was critical and unavailable components. When you develop a new product that is complex like ours, that has so many components, you have the instrument, the microfluidics, the reagents, the software, I mean, you have so much to do. You have to choose on certain points to focus your attention, otherwise you will take not 3 years but 10 to develop the product. And so starting with where do we put the different topics, for example, microfluidics, chemicals to do the microfluidic step that we needed, this was really what we invented. So nobody in the world knew how to do it, so that we had to focus on that, we have to do it ourselves. Then the PCR reagents, we were just doing PCR, I mean it's typical PCR, but it's just a PCR reaction. So we didn't try to invent a new enzyme, a new PCR chemistry, we just took many different PCR reagents that were available off the shelf, we tested it in our systems, and the one that worked we kept. The microfluidic instrument, the chip is something that's unique. We have to think of how will it plug into an instrument, so that is something we have to focus ourselves. But the PCR instrument, the fluorescence optical reader, I mean, these are just, standard stuff that were also, existing at the time. So we pretty much also screened different solutions and we bought them off the shelf and built our first products around those components. And it's a bit of the same story around the software, focusing on the analysis software and not the data acquisition software. And by really choosing our efforts, that's how we were able to go from, in a short amount of time, 3 years, from some really corner table microfluidic experiments to visual PCR into a product that we could sell and it could be used by hundreds of customers. And lastly, I think this is obviously always very key. The team has been crucial and the team has evolved a lot over the years. We can go through the slides. But I was always very keen on developing diversity in our team. So having people from different origins, men, women, obviously, but then also different nationalities, Canada, England, France represented in the founding team. And then later on, the team today still shares that diversity in terms of profiles, cultures, experience, and that's absolutely useful every day. In general, if we go to the very last slide, I think. Let's go to the very last one. I did that slide a few years back to think of all the different experience that we brought into the company by hiring new people. And I started myself, I only knew about physics, mechanics, microfluidics. I could talk about what I do, so communication, the rest I didn't know. And so every time I hired a new person, I was always thinking, am I bringing something unique to the company in terms of skills? And that's also something we still do today at Stilla. Even though we're 100, the 101 employee will have to bring something new. If that person doesn't, then it means, okay, it's redundancy, it can bring some security, but at the same time, there are still some zones of experience we don't have today and that we should probably take care of. And that's more important, in my opinion, than filling out all the blanks. All right, so that's quickly what we do, how we did it, sharing some retrospective learnings, but there are many, many more, so please, if you have any questions.